I have been implementing the next generation science standards in my classroom, specifically my 10th grade biology classroom is the example I used, and how I used Vernier technology to do this. 1 dimension and 3 dimension refers to are the 3 parts to the next generation science standards. The typical part that we normally think of are the disciplinary core ideas, and 2 pieces that next generation science standards added were the cross-cutting concepts and the science and engineering practices. For my example here, the content that I used was photosynthesis, and what I found was a lot of times my students were leaving my class not knowing any more than they did coming in about photosynthesis. I also challenged myself with adding the engineering practices into my teaching. I integrated the 3rd dimension into my classroom, and that is the cross-cutting concept, the energy and matter, flows, cycles, and conservation. So the lesson that I designed for this cross-cutting concept with the content of photosynthesis and using the practice of engineering, I designed a solar cell challenge where students made a model of a chloroplast, a working model, and this model could collect light and it converted it into energy. And to make this model of a chloroplast, we actually extracted plant pigments. This is a picture of what the solar cell looks like. So the first step here was they had to prepare the glass slide. And then the main step was design plant pigments to capture the full spectrum of light. And then they soaked their glass slides in the pigment that they designed, and then finally they tested their solar cells. In order to do this experiment, I also added the use of the SpectraVis Plus, and that's a product that's made by Vernier. And the SpectraVis Plus, if you put the pigment of the plant inside this cuvette and put it in the machine, this is the output you get on the Logger Pro program, and so it shows what wavelengths of light are absorbed at what levels. Then this is an example of what anthocyanin would look like, so you have a peak more in the green area. Their goal was to get all of the light colors absorbed to maximize the amount of wavelengths absorbed by their solar cell.
Because these 2 both have more color, it's going to take away the color from that one.
I have mine's lots of blackberries, some blueberries, and then algae and water. Everything. I feel like blackberries are best because they have lots of colors and they have and they're light but also dark.
We're trying to get as many colors in it as possible so that it turns black eventually. It absorbs a lot of blues and...
What do you want it to absorb more of?
The greens.
The yellows.
And
red.
And the red. So what are you going to add to make it absorb more red?
So you want this, right?
Raspberries, because they absorb...
No, they reflect.
So blackberries. You guys, maybe we should try...
But also if you add more green, they're going to absorb more red, because chlorophyll is green and it absorbs a lot of red.
Some more algae.
And we're going to go to the greenhouse pretty soon to get more of your green.
A little bit of everything. These things. This is dark.
We're really able to understand how plant leaves work better and how chloroplasts work better. And then they had questions afterwards like, why aren't plant leaves black or dark, really a dark color? Because that's what pigment would absorb the most colors. So we talked about problems with that and cell damage if they absorb too much energy, and so there had to be a balance. So to sum everything up, this was a really successful engineering activity that we did, and it would not have been possible to add the engineering piece if we would not have had access to the SpectraVis Plus. So that was one way this year that I integrated the next generation science standards in engineering using Vernier technology.